Hi everyone, this is going to be our lecture on chapter 15, which is our special senses. This lecture will probably be divided into a few video segments because, like several other of our lectures, this PowerPoint is very long. So to help break it up for you and for me, we will split it up a little bit. Let's go to screen sharing mode and check out what your book has to say about special senses. First, we have special sense receptors. Our special sense receptors are providing important information about our environment. There are two types of receptor cells involved in our special senses. You have dendrites of our specialized neurons, like our olfactory receptors. They will depolarize when they're contacted with a specific stimulus, like a dissolved chemical, if it's an olfactory receptor. Here you see what I mean. Stimulus comes in, stimulates our specialized dendrites of our specialized olfactory neuron, and that causes that depolarization, which is called a generator potential. The generator potential has to exceed threshold, just like in a normal action potential where threshold has to be met. You also have inexcitable membranes. So these are going to form synapses with the process of sensory neurons. These, when they are depolarized, they will undergo a graded depolarization. They will trigger a release of chemical transmitters at the synapse, and they will depolarize the sensory neurons to induce that generator potential. So the action potentials will then be propagated to the central nervous system, and there is a slight synaptic delay with these specialized cells. They also allow for modification on the receptor cell by having some type of presynaptic inhabilitation or facilitation. So taste, vision, equilibrium, and hearing all use these inexcitable membranes as well. Here you see what I mean. There's a receptor cell that has to be stimulated before the axon of the sensory neuron is stimulated. Again, this receptor cell can either be excited or inhibited depending on the stimulus. You can pause the video here and go back and review these three questions. Let's start with our special senses with the discussion on olfaction. Olfaction is our sense of smell. It uses our olfactory organs. Our olfactory organs are paired structures that provide olfaction for our enjoyment or displeasure, depending on what you're smelling. Located in the nasal cavity on both sides of the nasal septum, covers the interior surface of the cribriform plate of the ethmoid bone, superior portion of the perpendicular plate, and the superior nasal concha of the ethmoid bone. The sensory pathway to the cerebrum starts with the olfactory bulb. The olfactory epithelium contains axons that collect into 20 or more bundles, which then go into the cribriform plate, and then they first synapse with that olfactory bulb, as I said. That olfactory bulb is going to leave, going back to the brain through a tract called the olfactory tract. And then it goes to the olfactory cortex, the hypothalamus, and portions of the limbic system. Like we've talked about in our sensory pathways discussion, that olfactory tract is relayed through the thalamus to the hypothalamus, olfactory cortex, and the limbic system, which is why our sense of smell is so closely linked to emotion, memory, and as well as some reflexes like hunger and salivation, stuff like that. You smell popcorn and you start salivating because you know that you recognize that smell as popcorn and it makes you hungry, and therefore all of your digestive responses are excited. Extended distribution explains why some of our smells have such a profound and behavioral response. Pretty much what I just said. So here you see all of that goodness of our olfactory sense. You see the olfactory receptor cells, all the bundles located within that ethmoid bone, the olfactory bulb, the olfactory tract leading back to the thalamus, and within the thalamus it's going to be rooted to all of those various structures that we mentioned. So let's talk about the olfactory organs. It contains the olfactory epithelium, which is lined with olfactory receptor cells. These are modified neurons. Each cell forms a little knob projecting past that epithelial surface into mucus. The mucus is covering that epithelium. There's about 20 million receptors in a very, very small space. There's also supporting cells and basal cells or stem cells, which are constantly being reproducing new receptor cells. This is one of the very few examples of neuronal replacement. There's also the lamina propria. So you have your olfactory epithelium and then the lamina propria on which the olfactory epithelium rests. This is areolar tissue. This is where numerous blood vessels are located as well as nerves. It contains the olfactory glands, which are called Bowman's glands. These secrete water and form that thick mucus that we associate with our nose. There you see them. Here's the gland embedded in that epithelium, the lamina propria, areolar connective tissue, and of course our olfactory epithelium with the olfactory dendrites being held in place by that mucus. Odorants are dissolved chemicals that stimulate those olfactory neurons. They interact with the membrane receptors, and they're called odorant binding proteins, and they bind to the membrane surface. They're generally small, inorganic or organic molecules, and the strongest smells are associated with molecules of either a very high water or a very high lipid solubility. So the ones that are neutral don't typically have a strong smell to us. There are 4 major odorant molecules that can activate a receptor. So the olfactory reception process occurs on the dendrites. The odorant binds to its specific receptor, activates adenylate cyclase. That's the enzyme that converts ATP into cyclic AMP or CAMP. The CAMP then opens the sodium channels within the plasma membrane, and that begins the whole process of depolarization. Sodium channels open, sodium rushes in, causes that membrane potential to become more positive, etc. If the depolarization is sufficient threshold, the action potential is triggered, and that information goes on up to the central nervous system. Here you see the binding. Our ATP is then converted into CAMP once that odorant molecule binds. CAMP is then going to cause our sodium channel to open and ions to enter. Our membrane can now be depolarized. You can pause the video here and go back and answer questions on this section. We have now finished our first special sense, and we're moving on to our second special sense, gustation. Gustation is the sense of taste. It provides information about foods and liquids that we eat and drink. Our taste receptors, or gustatory receptors, are the most important ones, the most important of which are distributed over the superior surface of the tongue. But there's also taste receptors on our pharynx, inside of the cheeks, epiglottis, so even back in our throat. And the numbers of these taste receptors declines with age. The lingual papillae are the projections on the surface of the tongue, and there's 4 types. There's valate papillae. These are the very large ones. They're shaped like the tip of a pencil eraser. They're surrounded by deep epithelial folds. They contain up to 100 taste buds each, and they form a V shape on the posterior part of the tongue. The foliate papillae are found on the lateral sides of the tongue, and they contain taste buds. The fungiform papillae are shaped like little buttons, and they contain about 5 taste buds each, and they're found on the anterior 2/3 of the tongue. And then the filiform papillae, which provide friction. They don't have taste buds on them. They're found on the anterior surface of the tongue as well. That's more for grasping and moving the food rather than tasting it. So here you see what we are talking about, the valate papillae, nice and circular, lots and lots of taste buds associated with them. Foliate papillae with just a few, fungiform papillae, again with a few taste buds, and filiform papillae with no taste buds. Let's talk about these taste buds. Taste buds are special sensory receptors. Each adult has about 5,000 of them. They're made of a gustatory cell, which is our special taste receptor that extends through a very slender microvilli through something called a taste pore, and the typical taste receptor cell lasts about 10 days before it's replaced, and there's about 40 to 100 receptor cells per taste bud. There's also basal cells within our taste buds. These are the stem cells which will divide and mature to produce more gustatory cells or more taste receptor cells. So there you see them, the transitional cells. Here's the taste hairs of the microvilli, and then the actual basal cells below, which are going to divide into these gustatory cells. The pink ones are the gustatory cells. Our taste sensations are divided into categories as well. They're sweet, salty, bitter, and sour. Our taste buds are in all portions of the tongue, providing all 4 sensations. They're sensitive to different sensations, may vary along where in the tongue it's found, but there's really no difference in the taste bud structure for these 4 primary sensations. There's some other sensations. Umami is a savory taste, like beef broth or Parmesan cheese. It's kind of hearty tasting. It's detected in receptors that are sensitive to a particular amino acid and nucleotides, and those receptors are present in the Foliate Papillae. There's also water receptors, which are concentrated in the pharynx or the throat. This information is sent to the hypothalamus, and it affects water balance and blood volume regulation. Overall, it's to help the over-ingestion of water. Then you have taste receptor sensitivity. So our taste receptors are actually more sensitive to unpleasant stimuli. So if you think you're losing your sense of taste, taste something like an anchovy or something that you don't like to eat, and you will taste it. It's 100,000 more times more sensitive to things that are bitter, or 1,000 times more sensitive to things that are sour compared to things that are sweet and salty. Typically, people like sweet and salty things and not sour or bitter things. So it's a survival value. It's a protective mechanism for our body. Toxic compounds are often bitter. Acids can create burns. So the overall sensitivity to these taste sensations declines with age as our number of taste receptors declines and the number of olfactory sensors decline. So you can pause the video here and answer these 3 questions. Let's talk about how gustation is received or perceived. First, you have to be stimulated by dissolved chemicals, much like olfaction. The chemicals that are contacting those little taste hairs can diffuse through the plasma membrane or bind to receptor proteins of the receptor cell. About 90% of those receptor cells are responding to multiple taste stimuli. So as we said, taste like salty, sweet, sour, bitter, etc. can all be tasted or sensed through the same types of taste buds. Different tastes will involve different receptors though. So the receptor cells will adapt slowly. Central adaptation reduces sensitivity to new tastes. So in other words, the first bite of that salt and vinegar potato chip, you're going to say, ugh. But then halfway through the bag, you're enjoying them. They're salty, they're tasty, they're crunchy, they're awesome, right? That's adaptation. So let's talk about the 2 types of gustatory receptors. You have salt and sour receptors and bittersweet umami receptors. Your salty sour receptors. Hydrogen ions or sodium ions diffuse through sodium leak channels, and the membranes of the cell is depolarized, stimulating neurotransmitter release. For bitter, sweet, and umami, the binding of the sweet, bitter, umami stimuli is going to activate G protein complexes called gustducins. Those activate a secondary messenger, which is going to stimulate the release of neurotransmitters. The result of the neurotransmitter is going to be the stimulation of the sensory neurons. The sensory neurons are depolarized, which leads to that generator potential and propagations of action potentials along the gustatory pathway of the central nervous system. So here you see what we mean, some type of taste sensation coming in, a salt sour or a bitter sweet, opening going into that resting membrane. That's ultimately going to cause a depolarized membrane because of that influx of sodium ions or hydrogen ions. Or it could cause the activation of a G protein, which is going to activate a second messenger to cause the release of neurotransmitters. Either way, that plasma membrane is going to be depolarized, and action potential will occur as long as the stimulus is strong enough. Let's talk about that gustatory pathway. So the receptors will respond to stimulation. We just mentioned that. That information is relayed through the cranial nerves, facial nerve 7, glossopharyngeal nerve 9, and the vagus nerve 10. I'll go up into our brain. The sensory afferents will synapse on the solitary nucleus of the medulla oblongata. Postsynaptic, and that's again a protective measure. So it's going to the medulla oblongata. Medulla oblongata is our basic functions. So it's there to kind of cross-check and make sure everything's safe before it goes up to the higher levels. So postsynaptic axons will cross over, enter the medial lemniscus of the medulla oblongata, and then go up to the thalamus, where it's going to the gustatory cortex, which is within the insula. So here you see the steps: gustatory receptors, cranial nerves, solitary nucleus, medial lemniscus, and then the gustatory cortex. All the same stuff, just on different slides. Let's talk about how taste is perceived. Conscious perception of taste is produced by the primary sensory cortex. Information from taste buds is actually correlated with other sensory data as well, like the texture of food or like the temperature of food. Imagine eating a cold cheeseburger, right? Might not be as savory as eating a nice hot cheeseburger with the cheese dripping off of it, right? Or a toasted bun, right? Buns taste the same whether they're, warmed up or cold, but it's just that temperature that we like. So texture of food, how many of you don't like the taste of something because you don't like the texture? People don't like mushrooms because they can be mushy, or some people don't like fish because, it has its smell to it or whatever. That's all in the perception. Olfactory stimulation plays a huge role in how we taste, several thousand times more sensitive than our taste is. So it's there as a protective measure as well. So if you want to eat something that you don't like, or you want somebody to eat something that they don't like, hold your nose. Remember that old trick? Just hold your nose and eat it. All right, you can pause the video here, go back and review gustation. We're going to move into equilibrium and hearing. These are the learning outcomes, which are already covered in your chapter or weekly folder. So let's compare. We were just talking about chemoreceptors. Chemoreceptors of olfactory and gustatory pathways, those are responding to dissolved chemicals and causing some type of creative potential reaction, action potential route to the CNS, right? And this is just a review, our mechanoreceptors are what our hearing and equilibrium are. They're isolated and protected from the external environment because you don't want them to be disturbed unless the stimulus is strong enough or unless the stimulus is actually present, right? So these are located within the inner ear. Information is then integrated and organized before it's forwarded to the central nervous system. It begins with these internal ear receptors called hair cells. Hair cells have a free surface that's covered with a specialized process called stereocilia, which is why we call them hair cells. They kind of look like little hairs. They contain a large kinocilium, which is kind of like a super big cilia, and neither of them are actively moved. So the mechanoreceptors are very sensitive to contact and movement. External force pushing on them is going to distort the plasma membrane, altering neurotransmitter release. It also provides information about the direction and strength of the stimulus. So it's always surrounded by supporting cells, dendrites of sensory neurons. They're well isolated and well protected. So here you see that kinocilium, lots of stereocilia. They're all bound in this extracellular fluid, and they're within these inner ear structures. They're connected directly to dendrites of sensory neurons, and supporting cells hold them in place. So let's talk about these receptors. They are very three-dimensional structures, and basically the structure of this kind of three-dimensional space is going to determine which stimuli can reach those hair cells in each region. So hair cells in one region are only going to respond to gravity or acceleration. Hair cells in another region are only going to respond to rotation or sound. So they're all specialized and compartmentalized based on the category of movement or sound that they're responding to. You can pause the video here and go back and answer these 3 questions. Let's talk about the anatomy of the ear before we get into too complex, receptor transmission type stuff. The external ear is the visible portion. The auricle is that elastic cartilage that we can see. The external acoustic meatus is the passageway through the temporal bone. There's skin lining the passageway. It contains ceruminous glands, which secrete cerumen, aka earwax. And there's little hairs in there as well to trap debris. Cerumen protects against the entering of foreign objects, bugs, and microorganisms. Ew, bugs crawling into your ear. Kind of gross. The middle ear is an ear-filled chamber. This is bordered by the tympanic membrane, which is a thin, transparent sheet. It separates the external ear from the middle ear. And there's also auditory ossicles or ear bones. The malleus, the incus, and the stapes. These connect the tympanic membrane to the receptors on the inner ear. The inner ear is where our sensory organs for hearing and equilibrium are located. They receive the sound waves from the middle ear, and they have contours which are established by bone. So in other words, the temporal bone is kind of chiseled out to fit like a puzzle piece our inner ear structure, and that's called the bony labyrinth. So here you see the bony labyrinth. It's a little nest that our inner ear structure sits in. External ear, middle ear, malleus, incus, stapes, our inner ear here with all of its crazy shape sitting in that bone structure, and then our major nerves, which are responsible for detecting equilibrium and sound. So let's talk about some of the details of the middle ear. Outer ear is pretty straightforward, right? It's just the pinna or the auricle, right? The auditory ossicles join are the tiniest joints in the body, and they are synovial joints. So, you know, they have little ligaments attached to them and things like that. The malleus is the hammer. This attaches to different parts of the tympanic membrane. The incus attaches the how come I can't say it already? The malleus or malleus, oh my gosh, to the stapes, and then the stapes is bound to the oval window of the cochlea. Malleus, incus, and stapes. How come I just forgot how to say it all of a sudden? Sometimes I go crazy. All right, as I'm just talking to myself here. Let's talk about some of the other details of the middle ear. You have the auditory tube, or the eustachian tube, or the pharyngotympanic tube. This is the little tube that drains your middle ear into the back of your nasopharynx. It allows for equalizing pressure on either side of the tympanic membrane. It also allows for microorganisms to invade and cause infection, like otis media or an ear infection. Your middle ears also have muscles associated with them. There's the tensor tympani muscle. This connects the malleus to the temporal bone. It can contract and stiffen the tympanic membrane, reducing the amount of vibration that you're feeling or sensing. It's innervated by the motor fibers of the mandibular branch of the trigeminal nerve, cranial nerve number 5. There's also the stapedius muscle. This connects the posterior wall of the middle ear and reduces the movement of the stapes at the oval window. Here you see some of these little muscles holding these bones in place. So, our ear is ultimately primed for amplification and protection. Sound waves vibrate the tympanic membrane and convert those sound waves into mechanical movement. The ossicles will conduct those vibrations into the inner ear. It basically functions as a lever system, collecting the force of the sound waves and causing it to be focused and concentrated on that one part of the cochlea we call the oval window. So the result is considerable amplification. There's not necessarily good for loud sounds. As we know, listening to loud sounds can damage our ears, but contractions of the tensor tympani, stapedius muscles will help protect the tympanic membrane and the ossicles from very noisy conditions. You can pause the video here, go back and answer these 3 questions. The bony labyrinth, remember, is the little nest within the temporal bone that all of this inner ear structures kind of live. It's a dense shell of bone. It surrounds and protects the membranous labyrinth. The membranous labyrinth is inside the bony labyrinth. Between the bony labyrinth and membranous labyrinth is perilymph. It kind of resembles the cerebrospinal fluid, and again, it's between the bony labyrinth and the membranous labyrinth. There are 3 parts to this area. You have the semicircular canals, the vestibule, and the cochlea. So here you see what I'm talking about. This brown stuff is literally the bone kind of case that this structure sits in. So it's this little case that our cochlea, semicircular canals, and vestibule sit in. It's called the bony labyrinth. The vestibule, semicircular canals, and cochlea are called the membranous labyrinth. It sits inside the bony labyrinth, and in between the two is the perilymph. Now, that membranous labyrinth is all fluid-filled tubes, and those fluid-filled tubes are full of endolymph. Endolymph is basically the fluid that is inside our inner ear. Its receptors, the receptors within the membranous labyrinth, are only going to respond to a specific stimulus. So they're very specified. As I mentioned, certain ones are only going to respond to rotation, or certain ones are only going to respond to acceleration or sound or whatever. So they're very, very specialized. So let's talk about this membranous labyrinth in more detail. You have the semicircular ducts, which are within the semicircular canals. They're stimulated by rotation of the head. I just stimulated my semicircular ducts, right? The utricle and saccule. These provide sensations of linear acceleration or gravity. Whoa, right? That's acceleration or gravity when you're falling, right? That's all done in the utricle and saccule. Then the cochlear duct. The cochlear duct is sandwiched between a pair of perilymph-filled chambers. So there's two chambers within the cochlea, resembles a snail shell, and this is where our sound is perceived or received. So membranous labyrinth has our semicircular ducts, our saccule and utricle, and our cochlear duct for hearing. You can pause the video here, go back and review this section. So let's talk about the receptors for equilibrium. These are in our semicircular ducts. The three ducts, anterior, posterior, and lateral, are continuous with the utricle. They're filled with endolymph. Each contains an enlarged region called an ampulla. The area housing the receptors in that region is called the crista ampullaris. Kinocilia and stereocilia of hair cells are embedded within an area called the cupula. There's flexible, elastic structure that extends the width of the ampulla. So let's take a look at this. Here's our expanded areas. We call that the ampulla. These are our expanded areas. We call them the ampulla. Within the ampulla is a structure called the cupula. Our cupula is made of a part called the crista ampullaris, which has supporting cells, hair cells. Remember, hair cells are the special receptors in our ear. They have cilia and stereocilia and kinocilia. They're all there to detect movement. It's filled with endolymph, and it's all connected to a sensory nerve. So let's talk about these semicircular ducts in more specifics. When our head's rotating in a plane, one duct moves the endolymph, it pushes the cupula to the side, causing those hair cells to be distorted. Movement one way causes stimulation, the opposite movement is going to cause inhibition. So the cupula rebounds to normal position when the endolymph stops moving. This is how movement is detected. A horizontal no is going to stimulate the lateral duct. Nodding yes stimulates the anterior duct, and tilting the head from side to side is going to stimulate that posterior duct. So here you see in the three different planes of space how some of these ampullae will respond. The utricle and saccule are providing equilibrium sensations to whether or not your body is stationary or moving, right? Are you on a plane? Are you on a train? Are you in a car? Are you moving forward, or did your train just stop? Did your car just stop, right? It's connected by a slender passageway, which is continuous with that endolymphatic duct. It ends in what's called the endolymphatic sac, and these little sac projections go into the subarachnoid space. After being secreted into the cochlear duct, the endolymph returns into the general circulation at the endolymphatic duct. So the utricle and saccule contain hair cells, which are clustered in areas called maculae. The hair cells of the utricle project vertically, the saccule project laterally. So the hair cells are embedded in a gelatinous material called an otolithic membrane. So remember, in our semicircular canals, again, I'm backing up. In our semicircular ducts, our hair cells were embedded in a cupula, and they were contained within this endolymph viscous fluid. Now here, we're looking at cells that are being contained in a membrane. So here you see the same structure, just a different function, so there's going to be slight variability. In our saccule and utricle, these little swellings right here, this little bubble and this little bubble, we have our supporting cells, our hair cells, which in this case we're calling them maculae. The maculae have that cilia on them, projecting into the otolithic membrane. It's a very thick, jelly-like material. On top of that otolithic membrane are otoliths. They're little crystals. All of these maculae are connected to a hair cell, or are connected to a neuron, a sensory nerve fiber, and that is going to carry the information to the vestibular cochlear nerve. So any changes in position of the head causing distortion of those hair cells is going to send a signal to the brain. When the head's upright, the otoliths sit on top of the otolithic membrane. When weight presses on the surface, compressing the hair cells but not bending them. With a tilted position or with linear movement, gravity is going to pull those otoliths side to side, and that movement distorts the hair cells, and that's going to stimulate the macular receptors. So again, the otoliths kind of tumble, and that tumbling shifts the membrane, and shifting the membrane shifts the hair cells, and shifting the hair cells stimulates the receptors. So you can pause the video here, go back and answer these 3 questions. Let's talk about the cochlear duct, our receptors for hearing, all within the cochlear duct. So it's a long coiled tube filled with endolymph. It lies between the perilymphatic chambers. There's the scala vestibuli and the scala tympani. The scala vestibuli is surrounded by a vestibular membrane. The scala tympani is surrounded by a basilar membrane. So both ducts are interconnected at the tip of the cochlear spiral, and they create one long chamber. It begins at the oval window and it ends at the round window. The hair cells for hearing are within something called the spiral organ, or the organ of Corti, which is in the basilar membrane. So let's take a look at this. We have our oval window here. This is where the stapes is first kind of tapping as it's carrying the vibrations from the tympanic membrane, and those vibrations are going to travel down the cochlear ducts, and they can stimulate the membranes within our cochlea. If we look at a cross section, you see that there are different membranes. You have a basilar membrane and a vestibular membrane. The vestibular membrane is this kind of inner, like blue-like membrane, and the basilar membrane is this kind of yellowish type membrane. The scala vestibuli are the large holes, and the scala tympani are the kind of holes that are just beneath it. So they travel in a pair. It's a double membrane system there. And notice they're all connected to these nerve fibers, which all branch into that vestibular cochlear nerve. So the organ of Corti is the specialized surface which is within this cochlear duct system between the scala vestibuli and the scala tympani. There you see a cross section of the whole cochlear spiral. So let's talk about that organ of Corti a little more specific. The hair cells are arranged in longitudinal rows. They lack kinocilia. The stereocilia are in contact with the tectorial membrane that's embedded in the basilar membrane. Sound waves are going to cause pressure within the perilymph, and that's going to vibrate the basilar membrane. The stereocilia will then press into the tectorial membrane and be distorted. The more movement, the more hair cells are stimulated. The sensory neurons will relay that information through the spiral ganglion into the cochlear branch of the vestibular cochlear nerve. So here you see the whole process. The tectorial membrane becomes distorted from the vibration. That's going to be detected by these hair cells. The hair cells are going to carry that information to the nerve fibers. Again, the tectorial membrane is within the organ of Corti, which is between the scala vestibuli and the scala tympani. There you see him, just some type of pressure wave from the sound waves. You can pause the video here, go back and answer these 3 questions. Let's talk about the physiology of hearing. So the waves of pressure are conducted through a medium, right, like air or water. In air, the pressure waves create an alternating areas of compressed and separate air molecules. That's called a wavelength, right? It's the distance between the wave crests. Hearing is the perception of those waves, the perception of sound. So, sound pitch is one thing that has to be perceived. All sound waves travel at the same speed, you know, regardless of medium. In water, they travel at one speed. In air, they travel at another speed. But as the frequency increases, the wavelength is going to decrease. So the frequency is the number of wavelengths passing through a fixed point in a given time, or the number of cycles through a given time. It's measured in a unit called a hertz. Different frequencies are perceived as different pitches. So a high frequency, which is 15,000 or more hertz, is a high pitch, or a low frequency, 100 hertz or less, is a low pitch, right? That's what pitch is. So here you see, and all that is referring to is how quickly these sound waves are bobbing up and down. How big is their wavelength? So There's also intensity. Intensity is the energy of the sound wave. This is going to change as in the form of what we call amplitude. The greater the energy, the larger the amplitude, the louder the sound. It's measured in decibels. So amplitude is this, and that is how loud or how soft you're hearing that sound. This is a chart showing the intensity of representative sounds, like a quiet library soft whispers about 30 decibels. Something like a gunshot or a plane is 140 decibels. Notice that higher the decibels, some damage can occur. So, sound waves cause vibration. Energy sound waves is a physical pressure. Sound wave striking a flexible structure, like a tympanic membrane, is going to cause movement. So at particular frequency and amplitude, the object will vibrate at that same frequency. That's called resonance. So the tympanic membrane is going to resonate with the sound waves, generating movement of our ossicles, which is going to generate movement at the oval window. That basilar membrane is going to resonate at different frequencies as well. So the stapes pushes on that oval window. Inward movement causes distortion of the basilar membrane toward the round window, and the opposite action happens when the stapes moves outward. The flexibility of that basilar membrane is going to vary along its length. Different sound frequencies affect different parts of the membrane. So the location of the vibration is referred to as pitch, and the number of stimulated hair cells is our volume. So something that is, if this is a crude drawing of our cochlea with our basilar membrane here, here's our oval window. Something that has a very, very high pitch is going to be detected very early in our basilar membrane, as opposed to something that has a very or relatively low pitch, right? Or something that is going to distort our basilar membrane is going to cause that round window to be pushed outward or inward, depending on which direction. So, overall events involved in hearing. Sound waves arrive at the tympanic membrane. Movement of tympanic membrane causes the displacement of our ossicles. The movement of the stapes pushes on the oval window. The pressure distorts the basilar membrane on its way to the round window, and the vibration of the basilar membrane causes the vibration of those hair cells to move against the tectorial membrane, and that information gets sensed and relayed to the central nervous system, all collected through cranial nerve 8, the vestibular cochlear nerve. You can pause the video here, go back and review. Let's talk about equilibrium or balance. This is all done through our vestibular cochlear nerve as well. The hair cells in the vestibule and semicircular canals become stimulated. The sensory neurons in the vestibular ganglia all carry information through the vestibular branch of the vestibular cochlear nerve, and the vestibular nuclei integrate that information from both ears and relay it to the cerebral cortex, cerebellum, the motor nuclei, the brainstem, and the spinal column. So there's also a motor response, like automatic movements of the eye. This is all directed through the superior colliculi of the midbrain. It keeps your gaze focused on one particular point instead of moving, right? The distribution of motor commands through the motor nuclei of cranial nerves can control the eye, the head, the neck, all those kind of head movements. That's through cranial nerve 3, cranial nerve 4, 6, and 11. Our motor responses include the vestibular nuclei relaying information to the cerebellum. Again, that's all of our coordinated efforts, right? Information can then be relayed through the vestibular spinal tracts of the spinal cord and adjusting peripheral muscle tone, and complements any of the reflexive movements that our head and neck may be doing in response to equilibrium and balance, like catching ourselves or making sure that we pick up our feet a little bit more if we see a log or something like that. So hair cells in the vestibule and semicircular ducts monitor our position. That information goes to the vestibular branch. Vestibular nuclei and the medulla oblongata relay the information. Superior colliculi detect any automatic eye movements. Cranial nerves involved are the head of the eye, neck, and head are involved. And the vestibular nuclei relay information to the cerebellum. And the descending tract is going to carry out information to the peripheral muscles. Awesome. This is all going through the functions of the vestibular nuclei. And in terms of hearing with the vestibular cochlear nerve, those hair cells in a specific area need to be stimulated. The sensory neurons relay the information through the cochlear branch of cranial nerve 8, and that information reaches the cochlear nuclei of the medulla oblongata, ascends to the superior olivary nuclei within the pons, and the inferior colliculi of the midbrain. And then the inferior colliculi of the midbrain coordinates the auditory reflexes that are involving the skeletal muscles of the head, the face, and the trunk. So you can listen to things and say, right? You can have facial expression, or you can move towards the sound or turn your head towards the sound or whatever. And ascending auditory sensation synapse at the medial geniculate nucleus of the thalamus. Finally, those projection fibers relay information to the auditory cortex of the temporal lobe, where you can perceive the pitch as well as perceive the volume. Here's the whole process. Stimulation of hair cells, activating the sensory neurons. That information travels to the cochlear branch of the vestibular cochlear nerve. From there, the cochlear nerve is going to communicate with the nuclei within the medulla oblongata, relaying that up to the pons in the midbrain, coordinating any reflexes associated with that, communicating that to the thalamus, where any emotion or any other sensory input needs to come into play, because again, the thalamus is our relay center. And then finally, all of that's going to the temporal lobe, which is where our primary auditory cortex is. Most of our auditory information from one cochlea is projected to the auditory cortex of the opposite side, just like visual, just like everything else. Some information from the cochlea on the same side is received by this auditory cortex. So these interconnections aid in localizing sounds from left to right and reduce the functional impact of damage to a cochlea or ascending pathway. You can pause the video here and review these 3 questions. I'm going to stop this video here because our next little section is covering vision, and that's a whole nother ball of wax. No pun intended. We are just done talking about the ears. Hopefully you found this video helpful, and I will talk to you guys later.